Hello, everybody. Welcome back from lunch. Hope you enjoyed our tasty, healthy foods. My name is Wes. For those that don't know, I have the honor of introducing our first short talks session of the conference. We have 5 great talks for you today. Each one's going to be 8 minutes long with 2 minutes for questions. If you are a speaker, keep your eye on the time. Look down at me below. I'll give you a little hand wave when you're at 7 minutes, so you know you only have 60 seconds left. And we will try, once we get to about 8, a little bit, we don't have a lot of time because there's a lot of other speakers, so we might have to cut you off. So try to be mindful of that. And without further ado in that, one last thing. If you do have questions, please do go to the microphones on both sides of the room so people online can also hear your question and we don't have to repeat it, and it saves more time. All right, with that, we'll go to our first speaker from the University of Rochester, Hannah Swan, who will be talking about approximate Bayesian computational approach to denoising miRNA sequence level count data. After each one.
Can everyone hear me? Yes. Okay, cool. Let me make sure I can work this mouse. Okay, so I'm going to be presenting what I've been working on for my dissertation thus far. I am currently a rising 5th-year PhD student at the University of Rochester in the Department of Biostatistics and Computational Biology, and this is an overview of one of the chapters of my dissertation, which is an approximate Bayesian computational approach to denoising microRNA sequence level count data, and I'm going to do my absolute best to get through this in 8 minutes. So, just a brief overview. I'm going to give a very brief introduction of microRNAs and isomiRs. I don't know if you guys have looked at the schedule, but there are other members of my lab here, and we're about to have a very microRNA heavy next half an hour or so need to give a little bit of context to that. I'm going to give an overview of our approach for denoising length variant isomiR level count data, and then end with some preliminary results. So first, I want to very briefly talk about the definition and function of microRNA. So microRNA is a subclass of RNA molecules. They are small, non-coding RNAs. They're about 18 to 24 nucleotides in length. And although they are non-coding, they have important gene regulatory function. And the way in which they work, an overview of that is that they, through complementary base pairing, can target a messenger RNA, and they can destabilize that messenger RNA or repress translation, and through that mechanism, control gene expression. So when we observe these things, we generate counts of sequence level data, and we can classify the different types of sequences that we see. And so we can have an observation of the canonical sequence. The canonical sequence is thought to be the true sequence, and that's going to be the sequence that you see correspond to a given microRNA in a given microRNA database. There's 2 main databases. Those are miRGeneDB and miRBase, and for everything I do, I use miRGeneDB, although miRBase is also pretty popular. And so then we can have some sequences that differ by 1 to several nucleotides from this canonical sequence. So they're going to map to the same microRNA, and they may or may not be true biological specimens. And these isomiRs are classified based on the changes that you make to the canonical sequence to derive that sequence. So first here we have 5' isomiRs, and those differ in length with the nucleotides being added or removed from the 5' end. We have 3' isomiRs. Similarly, those differ from the canonical sequence by adding or removing nucleotides from the 3' end. A polymorphic isomiR is going to be the same length as the canonical sequence, but have substitutions along the sequence itself, which are shown here in red. And then we can have a mixed type isomiR, which is a combination of polymorphic and 5' or 3' isomiRs. And those both differ in length and have different sequence. And so what I'm going to be talking about here today is what I call length variant isomiRs, and those are both 5' isomiRs and 3' isomiRs. And so when we are working with the count data, if we have counts of these length variant isomiRs, they may have arisen from a biological phenomena in the cell and have actually been synthesized throughout the cell the synthesis pathway, or they may arise due to errors in the sequencing process, and therefore they can dilute the signal when we're doing a differential expression analysis at the sequence level. And so we want to make the decision whether or not to remove these sequences and their counts from the data and absorb them into a different sequence, or keep them because they have biological function. And so there's some key assumptions to the error model we're going to fit that allow us to do that. The first is that the formation of these length variant isomers is a rare event. The second assumption is that formation of 5' and 3' length variant isomers from the same reference sequence is an independent event. The third assumption is under the null hypothesis, the only source of length variant isomers is technical variation from the sequencing processes. So under the null hypothesis, only errors in sequencing are giving us these length variant isomers, and we can model that process and model the rate of formation of 3' and 5' length variant isomers using independent binomial distributions. So within a given sample, the observed read count of the 3' length variant isomer differing by n bases from the canonical sequence is going to be binomially distributed with these parameters, and then we are going to perform inference on these binomial parameters. So this is an overview of the computational approach. I have condensed like many slides of mathematical notation into this one slide. So if you have any questions or comments, I have a slide at the end that has like my contact information, so please feel free to reach out. So we're going to begin with the sample of observed isomer level read counts. And these read counts have been generated via alignment using merge. And then for each microRNA in the sample, we identify the most abundant sequence in the sample. We're going to call that the reference sequence, which is a proxy for the canonical sequence, but it's not the exact same because for reasons with the biology, the canonical sequence is not always the most abundant, but we want this to be independent of database, so we're going to do it this way. Then for each microRNA in the sample, we're going to identify the sequence and associated read count of the n-base 3' and 5' length variant isomers. And this is where the approximate Bayesian computation approach comes in. So we're going to use the observed read counts and the assumptions from the previous slide. We're going to generate draws of the independent binomial distribution probability parameters. And using an approximate Bayesian computation approach, we're going to perform posterior inference and then generate draws from the posterior predictive distribution of draws of read counts of the length variant isomers we may have observed given the data. And then we're going to compare the observed length variant isomer read counts for each microRNA to the read counts expected under the error model. And the idea is if only a very small proportion of drawn read counts are greater than or equal to what we observed, then that read count of that particular isomer is too abundant to be explained just by our error model. And so I have some preliminary results of this proposed algorithm that we are used to denoise experimental benchmark data that we have. So this is from the ERCC data, which is from the Extracellular RNA Communication Consortium. And the idea here is that it's all synthetic microRNA data. So we have no signal. And so we can use that to get an idea of our false positive rate, but also artificially add signal and make sure the method is working as we would expect. And so here we have 18 samples. We have the true positive rate of correctly identifying biological length variant isomers, the false negative rate, the false positive rate, and the true negative rate. And the key takeaways here are to focus on the false positive rate column and the true positive rate column. And we can see here that the false positive rate is at or below 0.05. And since we're doing this at a 0.05 significance level, that shows that it's working as expected, although it is perhaps a little bit conservative. And then on the true positive rate column, we can see that there's a range in the power of our method to detect these true biological isomers. And so this suggests that I might need to play around with my prior distribution specification, or it could be dependent on the sample itself with the library size. But overall, promising results, so we're going to continue working on this. And these are just a couple references that I have. This is the reference for miRGeneDB. This is where my figure came from, and then, or this is where my figure came from, and then this is another reference. And if you have questions or comments and we don't get to them during this session, this is my email. And you can also find me on LinkedIn. And like I said, I'm currently working on this for my dissertation, so any feedback would be greatly appreciated.
So my question's focused on your assumptions. You assume these are rare events. How rare is rare?
I don't know off the top of my head, but rare as in it comes from the literature surrounding the sequencing technology. So I think it's like 0.5% for the error rate.
So you're basically off of the sequencing technology, like actual error rate.
Yeah.
Gotcha.
Yeah.
Yeah, I wanted to ask you about the false negatives. What do you consider a false negative?
So a false negative would be if we artificially added signal to 20 of the 1 base 3' length variant isomiRs, for example. And so we inflated that read count so it would be really abundant. And if the model missed it and said that it was just a technical variant instead of a biological variant, that's a false negative.
Thanks.
Thank you.
Thank you.
And I do want to say this for future speakers. When I was up here originally, I said I'd be down there. That was a height bias, and I realized on all speakers can see over the podium to where I was sitting. So I will be over there by the screen now. Cool, because I'm tall and I didn't judge for that. Our next speaker we have is also from Rochester, is Ernesto Aparicio-Puerta. Oh, probably butchered that. I'm so sorry. Assessing differential expression strategies for small RNA sequencing using real and simulated data. Thank you.
Hi, everyone. Well, thanks, Anna, for introducing all this, because you're going to save me a bunch of time. Yeah, if you're not convinced already that it's pretty important to study microRNAs, it's probably one of the one kind of non-coding gene that is most studied. This is already a little bit old. It's from a paper a couple years ago, but you can see that the amount of sequencing experiments out there are increasing a lot. So it's important that we have methods that are specific to this, because basically people are calculating differential expression for microRNA-seq data just using regular RNA-seq. And I'm not going to go too much into it. Probably my 2 colleagues following me may. But there are a bunch of assumptions that these methods take that do not work for microRNA-seq. Yeah, so this is why we need these methods. This here in the middle represents kind of different things that can go slightly wrong and generate bias in your experiment. So you're never going to get microRNA counts that are proportionate to what you actually have in your sample. But let's pretend that we do. That's an assumption there. But even in that case, when you have changes in the absolute amount of one of the genes, for instance, here I did that on microRNA A, you can see that because you have to normalize somehow, and for microRNAs, we just normalize in counts per million. We don't take into account the length because they're all roughly the same length. You're going to observe changes in the other 2 genes, which are not there. This is normally not so much of an issue for RNA-seq because there's a lot of genes and the expression is not exactly proportionally distributed, but it's a bit more random. And that's definitely not the case for microRNA. There are a couple or a few microRNAs that will take much of the expression. So for these methods, like I said, my colleagues are working on developing these methods. I wasn't happy with the ground truth data sets that we had to evaluate them. Basically, there are 2 ways that we can generate ground truth data sets. And if someone is like an expert in this kind of thing, I would be super interested in talking to them after. But basically, you simulate them with some assumption and some model, or you sequence real data. But you need to know what's in that real data. So either you synthesize something and then you sequence it, and I'm not going to go into it, but that's not like a great idea. Or you can sequence real samples and then measure with a different methodology, but this is kind of circular. Like, how do you know that the other methodology is not throwing you off? So we kind of decided we want to merge these two parts in and come up with our own data set where we don't really simulate things, but we can control a little bit how we make two real samples, and that way we have some control, but still it's real data. So we came up with this design where we're going to mix at known ratios two different samples, different enough that we can pretty much tell all genes where they're coming from. And this is obviously not working 100%, but that's the idea. And because we know this, we have kind of our ground truth. And because we have real data, we can check how well our method is picking up or not these changes. So the theory here is we're going to be comparing from, from this blue comparison here to the red one and the green one. And as you progress down this list of comparisons, they will be more similar. So it will be harder to distinguish true differences, but that's the point of having like a benchmark data set. And yeah, I know I said I would use real data, but unfortunately, we're dependent. We are completely dry lab. We're dependent on someone providing the data, and we didn't get it by the time of the conference. So I'm only going to talk today about simulated data. And very quickly, the way I did it is basically the same concept as I proposed to the real data. I'm going to scramble a bunch of FASTQ files with known proportions, and yeah, just sample reads out of them until I reach always the same amount of reads, just so it's more or less consistent. Then I go back to the original files and I decide which ones, like very strict sort of threshold, which ones are true and without any reasonable doubt differentially expressed microRNAs. And then we can go back to the samples and make all these comparisons and check which of the methods are recalling these differentially expressed genes. And here at the left, you can see how, by the way, I'm comparing HRD-seq and lemma, which is what most people are using, but eventually I would like to test the methods of my colleagues and more methods, but these are probably the most well-known and most used in bulk sequencing. And you can see that for those comparisons that are a bit, in theory, harder, HR has a bit of an edge, no pun intended. And I was thinking maybe this has to do with how each method has a different correction, but actually not really, because it seems to hold anyway. I thought, okay, maybe HR is just calling a bunch of more genes. So of course, it will end up catching those are truly differentially expressed. But if you look at the fraction, it's actually doing even better. So, and again, I compared without multiple test correction. So yeah, I tried to look on why this is happening, if there are like tremendous differences, both in the expression of the genes that it can pick up or in the fold change. And it doesn't seem to be the case. I mean, it seems like it's better at picking up under-regulated genes. But yeah, this is just very preliminary. And yeah, this is just to show that we really need something to check like a ground truth to check our experiment. And that's my 8 minutes. Thank you.
Oh, sorry about that. Lovely talk. I noticed that your colleague used ERCCs as with synthetic 3' or 5' variants as a test vehicle. The ERCC sequences, the short spikes are similar length to microRNAs, or I remember whether they cover the range of both mature and pre-microRNAs or just microRNAs.
I think it's just microRNAs, mature microRNAs.
So one thought occurs: are there data sets out there that were spiked where it would be feasible?
Yeah, I think there are some data sets out there, but I have a slide here of why we don't want to use those, or we tried using those and they're useful in some context. But if you look here, this is what we refer to as the ERCC, some of the samples of the ERCC spikings. And as you can see, if you look at the top 50 microRNAs with the largest expression, the distribution is not really wide. So it's pretty much, they have some variance, but they really don't compare to some samples that I just picked from some real experiment here in orange.
So is that part of the reason why EdgeR is doing better? Is it estimating transcript-wise dispersion and capturing some of the true sequence bias heterogeneity better, or?
Could be. I think from the top of my head, the SEC, one of the assumptions is that it has same amount of true positives and false positives. Overexpressed and underexpressed. And I think for this data set, just the way I picked it and the samples that I chose, it didn't hold. So it threw it completely off. And then Lima, I think we know that it's a little bit worse. But the ESEC, I was expecting to be comparably good to EdgeR. But yeah, it could be.
Kind of exciting result. Thank you.
Thank you.
Yeah, I don't have a specific question. I just wanted to say that back in my PhD a while ago, I also did a small RNA-seq differential expression analysis, and there was no standards for it back then, but I also ended up settling on EdgeR as the best tool for it. So just to confirm your experience here.
Oh, thank you.
On real data.
I'd love to talk later.
Sure.
Our third speaker, I'd like to welcome up Seong-Hwan Jun. Am I saying that right? No. Oh, you can say it correctly.
Seong-Hwan Jun. Thank you. Okay, so so my talk is titled "Improved Differential Expression Analysis of microRNA-seq Data by Modeling Competition to be Counted." So I guess my colleagues have already established the importance of microRNAs. So they play a central role in regulating gene expression and modulating diseases. But the current practice in the literature is to apply methods developed for messenger RNA sequencing data, DSeq2 and EdgeR, which I'm pretty sure almost everybody in this room have used before at some point in their careers. So they work well, but there's some characteristics of microRNA-seq data that's not very well captured by these methods, and that's basically the topic of research here. So one of the key assumptions of the bulk mRNA-seq method is the approximate independence assumption. So basically, here when you have large number of features, in this case genes, and you have large enough input reach to be distributed to these features relatively evenly, then the correlation across these features will be small. In that case, you can sort of ignore them and do the analysis as if though, instead of doing multivariate analysis, you can actually look at them each independently. So this is a key assumption that underlies bulk mRNA-seq methods. And looking at the microRNA sequencing data, we noticed that they're substantially different. So for example, the number of features that you need to cover 90% of the reads for microRNA-seq is about 30 to 50, whereas for messenger RNA, it's usually well over 5,000. So there's way less features. And looking at the entropy also here, you can sort of see that the reads are more evenly distributed for mRNA-seq because the entropy is higher, so that means there's more uncertainty. Whereas for the microRNA-seq on top, it's smaller entropy, meaning the few features take up most of the reads. And the similar plot here shows that if you look at the median of the top 10 highly expressed features, like the top 5 features here take up like significant amount of the total input reads, compared to messenger RNA-seq on the right side, where they're more or less evenly distributed. And I noticed that roughly about 2 to 3% of the reads go to like the top 5 features each, so that's only about 10%. So the rest of the 4,000 features also account for quite a number of reads. And also looking at just the simple correlation plot, I noticed there are negative correlations between cell types in the microRNA-seq data. But in messenger RNA-seq, there's even positive correlation, or basically not much correlation between the top features. So the reason for this is because there's non-negligible competition to be counted among the transcribed mature microRNAs. So this means that the approximate independence assumption is likely violated. So we like modeling raw counts using negative binomial distribution because we can model over dispersion, and like we don't have to transform the data to normalization and check that the transformation worked and so on. So we want to keep using negative binomial just as DSeq2 and HR. So if you recall from DSeq2, basically they modeled the absolute abundance. So this SI is a size factor estimation, and QIJ is the absolute abundance of microRNA, or I guess for their purpose, it's the gene J in the sample. And then they fit basically thousands of regression models, and just basically they're looked at independently. So the simple modification that we propose is to actually model the mean expression μij as SI, the size factor, times pi ij, where pi ij represents the relative abundance of feature J or microRNA J in sample I. And the way we do that is using softmax regression. So we call this model negative binomial softmax regression. So doing so, there's a couple of advantages that come from doing this, and one is that we can now formulate fold change as a relative risk, which is a well-understood quantity in statistics. And the other thing is that the dispersion parameter can now be modeled in relation to relative abundance pi ij. If you recall, basically the biological coefficient variation, square of that is actually equal to dispersion. And the biological coefficient variation is a quantity that's related to the randomness of the pi ij. So this is more, so we're able to do more canonical modeling of the dispersion compared to, say, DSeq2 and HR. And some simulated results. So we are statisticians, so I just want to make sure that as you increase the sample size, the mean square error decreases, and that's the case here. The purple here is our method MBSR. And HR and DSeq2 also decreases, but not quite as good. So these are all very solid statistical methods. This research is not to bash this method. They're really excellent methods, but we're just proposing a simple improvement that seems to achieve high performance. And this is a 95% confidence interval. So again, as the sample size increases, we're able to recover reach the nominal coverage levels. And again, the error rates are much better as well. So this method seems to work. And about the dispersion parameters, so basically there's two underlying experimental conditions, and when there's a change in the underlying distribution over pi's, basically then you need to model the dispersion sort of like separately, and this model is able to do that, whereas HR and DSeq2, they are sort of estimating values that are somewhere in between. So the red and the blue lines are the DSeq2 and HR. And the X's are the true dispersions. And the purple is the MBSR. And so we've done some real analysis as well. So this is a plot of the immune cells, CD8 T cells and CD19 B cells with sample size equal to 15 for each cell type. And we noticed that one of the common practices in the differential expression analysis is using the thresholding on log fold change. So if the absolute fold change less than 1, you don't even look at. So, but if you do that sort of practice, then a lot of these highly abundant features will be ignored. Like, for example, the first one, miR1425P, that's fold change of 0.14, but it still picked a significant by our method. So these are the things that will be missed by, like, if you use like the more standard approaches. Yeah, and there's a further plots basically showing the dispersion estimation is needed for to capture the changes in the underlying distribution of pi. I missed is time okay, or what's, yeah, okay, cool. All right. So yeah, so the other thing, important thing is that when the sample size is small, so n equal to 3 for this cell line data analysis, DSeq and HR estimates an absurdly large fold change for some of these microRNAs, the red points here. So we looked at them, and most of them are like lowly expressed. So this is the MA plot. And a couple of them I noticed that were relatively largely expressed, but looking at them, they're mostly to do with the sparsity. So the DKO1 basically is unexpressed. And even for this one, miR47235P, there's quite a bit of sparsity. So there's 0 expressions in DLD1, DKO1, and DKS8. In these cases, DSeq and HR seems to not work well, whereas the MBSR seems to more graciously or gracefully handle sparsity. So the paper is up on archive. So if you want to take a look and if you have any comments, that would be really appreciated. Thank you.
Hi, this method looks really interesting. Well, it's developed for microRNA-seq. Do you think it would be applicable to like mRNA-seq or any other kind of data?
Yeah, so we're also looking at that, but I guess we're thinking about using it for single-cell RNA sequencing data as well. But there, the sparsity is more complicated because this microRNA-seq is, at the end of the day, is still bulk microRNA sequencing data. Whereas for single-cell RNA-seq data, there's more, there could be more things going on underlying that describe the sparsity. So it won't be straightforward, but that's definitely one avenue of research that we're interested in. Yeah.
Thank you.
Thank you. Yeah, thanks.
You guys brought the team bus from Rochester, eh?
We did. It almost went off the rails, but you know, we got here.
I'd like to introduce our next speaker, Andrea Baran. Thank you. miRglmm, modeling isomiR-level counts, improves estimation of microRNA level differential expression and uncovers variable differential expression between isomiRs.
Hi, my name's Andrea Baran. I'm here to talk to you about miRglmm, which is a method that we developed that models isomiR-level counts. And we're going to show you that it improves estimation of the microRNA level differential expression and can also uncover variable differential expression between isomiRs. And this is from a work from a recent preprint that I put up with these lovely colleagues. So my colleagues here did a bunch of the legwork of the background for this. MicroRNA are small RNA species. You already heard about isomiRs from Hannah. Typically, what is done is that isomiRs are aggregated to microRNA level counts, and this discards useful information. So the biological importance of isomiRs highlights the need for a microRNA-seq analysis method that can account for distinct isomiR expression patterns. So here is an example of isomiR level data. This is for a specific microRNA LET7A5P. The columns here are samples representing two tissue types. The first three are from bladder samples. The fourth, fifth, and sixth are from testes. And the rows here are the different isomiRs that map to LET7A5P. The first one is the canonical sequence, is what you would find in the microRNA databases. And then the rest of the rows are different isomiRs, length variants, or internal modifications are shown in red. The bottom row here is the aggregated microRNA level count. So this is calculated by just summing all the reads that map to any isomer that aligns to set or let 7A5P. So if you look at the overall microRNA level expression, there's no big difference between these two groups. But when you look at individual isomiRs, you can see that there's quite a bit of difference between some of these isomiRs between the two groups. Like some are not expressed at all in testes, but then have higher expression in bladder and vice versa. So we are losing information by just summing them all together. This is just kind of a visual representation of the same effect. So I have tissue-specific box plots here, which are summarizing the counts for 3 different microRNA based on the 3 different plots. So the aggregated counts are what are typically analyzed. So here you would see that testes is showing higher expression for all 3 of these microRNAs compared to bladder. The canonical, I'm just showing here for completeness, but I'm kind of gloss over that for now. But then if we look at the next 2 highest expressing isomiRs, you can see that there's a difference in the trend between the 2 tissue types. And when we aggregate, we kind of miss this effect. And so we want to know what happens when we account for this effect. So this is kind of the model that's underlying miRglmm. It's a negative binomial generalized linear mixed model based differential expression tool for microRNA-seq data. It's an R library that consists of 1 core function that can be easily integrated into differential expression analysis pipelines. And for each microRNA, miRglmm fits this model, which I'm going to kind of gloss over for the interest of time. But one important thing I want to mention is it's a negative binomial model, so it can directly model the count data without any needs for any sort of pre-processing normalization step. And also the effect of interest here is this beta. This is going to be like our log fold change estimate. So in order to assess the performance of miRglmm, what we did was we wanted to compare it to other commonly used differential expression tools that are limited to running on aggregated counts and cannot account for any sort of isomer level variability. We did this in a collection of monocyte samples. So we took a homogeneous starting sample set and we simulated differential expression at both the microRNA and the isomer level. So we picked a set of microRNA where we upregulated and then a set that we downregulated. And so we had a truth, and then we're going to use MSE and confidence interval coverage to compare to this truth. So mean squared, and this was done across 100 simulations. So we can see here that miRglmm minimizes the MSE and maximizes the mean coverage proportion, which is showing that we can better recover the true estimates and that our confidence intervals are appropriately capturing the truth. So when we actually look at this in an even more granular way, we can look at how well the methods are doing in the set of microRNA where I actually induced the effect. So for this group, these are the microRNA I didn't induce any effect into. The true fold change is 1. And then the other 2 groups are where I either upregulated or downregulated it. And you can see here that when there was no effect, DSeq is doing the best. It's got the lowest MSE, and miRglmm's right there with the rest of them. But really, it's where there was a microRNA level effect with the random isomer variability induced, where we really see that miRglmm is outperforming the other methods in terms of MSE and also drastically improving the confidence interval coverage proportion. So miRglmm that can account for isomer level variability provides consistently high performance whether or not there is significant isomer differential expression variability. And we think this is because, if you just model the aggregated microRNA level counts, you're losing information. So in conclusion, isomiRs are biologically important and can have variable usage between sample groups. miRglmm is a differential expression expression method that's developed for microRNA-seq data that uses a generalized linear mixed model of isomiR-level counts, which facilitates detection of microRNA with differential expression or differential isomer usage. In simulations, miRglmm provided a lower MSE and better confidence interval coverage proportion than other differential expression tools. And we find significant isomiR-level variability exists within most microRNA in real biological data sets. Kind of glossed over that here, but it's in the preprint, evidence of it, which further motivates the use of miRglmm to analyze microRNA-seq data. And then this is the link to the preprint. The package is up on GitHub, and my contact information. And I would like to acknowledge everyone else in my research group.
Hi. So my question is, the idea of isomiRs seems conceptually similar to the idea of alternative splicing in mRNA. So have you either tried methods for analyzing alternative splice forms and tried applying them to isomer counts, or have you compared it to that in any way?
No, we haven't yet. So that is definitely something that I could look into doing.
I'd be interested to see that comparison.
Yeah, thanks for the suggestion.
And that's all. Our last speaker for the session, from Grand Rapids, right here for a change up of location, we have Frederic Bertrand.
Okay, hello everyone. Thank you for the talk. We will not deal with microRNA, but with sensitivity analysis. So what is sensitivity analysis? So when you use numerical methods or models, how can I just go to the next slide? Okay, this one. So basically, the previous speakers design models to analyze data. And when you have your model, one of the things that you can do, and that I teach at university for about 10 years, is to use sensitivity analysis to retrieve information on the variables in your model. So the technique is well known. And one toy example, quite classical in the domain, is the J function. So the J function is the one that is defined this way. And basically, you have several parameters, the AA. And according to the value of the AA, from 0 to a large number, the variables are more and then less important in the model. So a plot of the function is this one. So basically, assume that you have a model that can be viewed this way. How can you find or compute the sensitivity of your model with respect to the variables? So basically, what you should all know since high school is some local sensitivity measurement. For instance, you have the partial derivative of the outcome with respect to the input at a given point. But it is more difficult to define something that measures global sensitivity. So basically, what you want to use with your model, when you have your model, you validate your model with the data, and then the model depends on some variables, and you want to know the sensitivity of the outcome with respect to one or pair of variables. Why? Maybe to carry out variable selection, or maybe just to say, define some criteria analysis. So you have several ways to use those tools. And basically, what we will deal with this talk are called global sensitivity analysis based on the variance approach. So those was designed by Sobol in the 90s and are very commonly used in numerical analysis or scientific computing. So what you get is some decomposition, so the Höfding-Sobol decomposition, which can be viewed as an ANOVA decomposition. So basically, you will try to decompose the outcome with respect to the income conditionally to some subset of variables. And there are several algorithms designed to estimate those quantities based either on Monte Carlo or quasi-Monte Carlo techniques. But what is interesting is that all those tools are only defined in non-random settings. And we all know that in biology and for omics data, for instance, we are dealing with models with random effects. So basically, what we did was to try to use the Sobol indices in context with random effect. So with the toy example, the G function, with two variables, we first computed the Sobol index for parameters that were set to 0, A1 is 0, and A2 is 1. And so we got Sobol index first order and second order. So second order is for interaction, first order is for main effect. So basically, you get the classical properties of Sobol indices, which should sum to 1 and should be between 0 and 1. So for the non-random setting, the use of Sobol index is already proved for a long time. But when you add random effect to your output, then it becomes quite ugly. As you can see, the Sobol index no longer sum to 1. And basically, the Sobol index are unable to grasp the fact that you have some random noise in your data. So this problem was already known and solved by the use of quantity of interest. So you have several references. And basically, what is the quantity of interest? It means that you will just fit your model a large number of time, and that you will use a summary statistic to summarize the quantity that you want. So for instance, the mean, median, or any quantile that you may want. So this works quite well for simple models with additive random effects. As you can see on the table on the right, you retrieve using the quantity of interest quite the same estimates that when we had not noised the response of the model. But as we saw in the previous talk, unfortunately, in omics data and for next generation sequencing, we mainly use Kant's model with Poisson or negative binomial and so on. And basically, as we will see later, it will not be as easy. So when you have a cover rate, noise dependent, a covariate that depends on the noise, the noise that depends on the covariate, it also works as soon as the effect is additive in the model. And so the work that we did with my colleague and the PhD student was to find settings and to explore what happens for Sobol indexes when you deal with uncertainties at the level of the distribution of the parameter of your distributions. And why did we do that? Because we had to study some stochastic processes, and it is also relevant for many models that are commonly used in omics data. For instance, generalized mixed model and Hurdle model, and so on. So as we saw in the previous talk. So in those settings, we use the toy example, which is that kind of process. So basically, you have two exponential distributions that are linked through a binomial one, and you have some uncertainty on each of the parameters of the distribution. And basically, you just measure the time that you need to get on the right branch of the process n times. And what we saw is that even with a very simple process as this one, you got completely impossible properties for the Sobol indexes that we computed. They no longer sum to 1, and they are even estimated as negative values. Maybe those. Okay, I stop. I missed the signal. So with Shapley value, it's not okay. Good too. And so if you want to perform sensitivity analysis, you have the classical packages in R, so Sobol and sensitivity, and the one that we will release very soon, Sobol for random values. Okay, thank you very much.
Hey, do you have intuition on why the random, when you have randomness, the Sobol indices don't sum to 1, and why when you take quantities of interest, like the mean or whatever, they start to sum again? I'm just thinking, is the mean much more deterministic than individual samples, and that's why you get back to the original deterministic case?
Yeah, in a word, you average out randomness. Yeah. So basically, you get a fixed effect model again, because
but why is it that in the first place you lose the summing to 1 quality?
It's because in the first place you have additive noise. So the mean is will just erase randomness this way. But with the other model, it's no longer additive.
Yeah.
So basically, you are just it's difficult to average it out. So
somehow the sum of the noise terms.
It's the problem in the general linear mixed model, that the average of the expectation is not the expectation of the average. So basically, there's something like that. You can look into it.
Yeah. Cool. Thanks.
Thank you.
Thanks, everybody. That concludes our first short talk session. We have a 10-minute break. You can stretch your legs, walk around. We'll meet back here, same room, same space for session 2.